Good morning, everybody. We're going to welcome on Dr. David Held. He is an Auburn University alumni professor and chair of the Department of Entomology and Plant Pathology at Auburn, and has done research on Crapemyrtle bark scale for a number of years, and he's going to tell us all about it today. So David, go ahead.
Great. I appreciate the opportunity to be with everyone and talk about something we've been working on for quite a number of years now. I think I'm on my third grad student working on Crapemyrtle bark scale. So looking forward to this opportunity to talk with you. So if we look at all the pests that are out there, potentially of major pests for Crapemyrtle bark scale among, Crapemyrtle aphid or even Japanese beetles, which I really love too, is probably the worst. It has really had the most negative impact on crapemyrtles throughout the South, more so than either of these other two pests almost combined in the time that they've been here. And we've had almost 100 years of crapemyrtle aphid in the Southeast. So you can kind of see the video on the side. This is kind of what a heavy infestation of crapemyrtle bark scale looks like. You can get these real heavy aggregations of these white, and they're called felt scales because they get this felt-like covering over top of the females and males that appear, and they very heavy on the trunk, on these outer branches, even sometimes in heavy populations, sometimes out there near the leaves as well. It came in through Texas and now pretty much is everywhere where crepe myrtle is grown. That doesn't mean every crepe myrtle is infested everywhere, but it does mean that there's a good chance that those crepe myrtles could become infested during the life of that tree. What does it mean to be infested? Well, at this time of year, when we're starting to get leaf flush, we've got the 60-mile-an-hour diagnostic of delayed leaf flush. So if you look at the picture kind of on the right, you can kind of see infested trees are much slower to leaf out. So as you're driving around in the spring, you might notice like, hey, that tree's, they're next to one another in a landscape, or those trees are slower to leaf out. That might be one of your first signs you need to get out and look at those trees in the landscape. Now, if we move on into the summer and you actually notice that, you're kind of seeing not as aggressive blooming out of a tree like, one of these white natchez or something, then that's another potential indication that the trees are infested. So there's just two timings of the year when if you don't feel comfortable, like, is this really a scale insect or what am I looking at? At least these are two conspicuous times of year when you can kind of get an idea if that tree could be infested. What does it mean to be infested? Well, that means we have a colony of these scale insects. These scale insects are plant sap suckers. And so they settle down in the crawler stage. And so if you look at this video, and sorry, it's so shaky, you can kind of see the little pink one moving around. That's a crawler. That's a crapemyrtle bark scale crawler. They come out from under the females. They hatch from the eggs. And we call them crawlers because that's the stage that disperses. So that's what we would call in bug nerd terms, first instar. They hatch out of those eggs. They crawl around, find a place to settle down. Some of them can be blown around in small areas by wind, but they have to settle down on a plant and start feeding. And the best plant forms are crepe myrtles. Now, what does that mean? What do we mean by crawler activity? These are the periods when we know we're having egg hatch, and it's the opportunity for this population to spread. Otherwise, once the crawlers settle down, they kind of attach themselves to the plants and they're not mobile anymore. So these are why these crawler activity periods are so important. Now, we know in Alabama, we've got, and really throughout the Southeast, crawler activity periods, usually September, October, are a real big peak. And then this window that runs basically from April into June. Now, those are the two main periods when we have crawlers. But what you can see from these data of where we were actually sampling crawlers on trees with sticky tape is that we do have crawlers throughout the rest of the year. I mean, we do have just a low level of crawlers that tend to hatch and be active on these trees moving around throughout a good portion of the year. But we know we've got the biggest activity of crawlers in this September, October window, and then April through June. One of the other things that can kind of tell you that you have crepe myrtle bark scale, but not specifically crepe myrtle bark scale, is high amounts of honeydew and sooty mold. One of the reasons I say not specifically crepe myrtle bark scale is because crepe myrtle aphids also produce a lot of honeydew. Honeydew is really just the sugary poop of sap-sucking insects. And we had a lot of false positives for crepe myrtle bark scale early on, because this is also one of the symptoms for crepe myrtle aphids. And so you'll see that shiny appearance. And then as that honeydew settles on things, we actually get the growth of sooty mold fungi, not only on the crepe myrtle, but as you can kind of see, even on some of the plant material that's underneath the crepe myrtles, will actually also have sooty mold. So just some other things to kind of give you an indication of the crepe myrtle bark scale activity and biology. They produce a lot of honeydew. It seems to be the most, in fact, we had an incorrect conclusion here, but it seemed like there was a lot being produced in the fall, like October. That made sense. We had a lot more crawlers, but what we come to find out from this work that was done by a student in my lab is that it's not that they're producing less honeydew throughout June, July, August, and September, but we have a lot of insects that are coming to these trees and eating the honeydew at that time. So something else that may happen is you may say, gosh, why does this Natchez crepe myrtle have so many wasps on it? Another one of those symptoms associated with potentially crepe myrtle bark scale. They could be feeding on that honeydew. And we do see high numbers of wasp flies and even lady beetles. And I'll talk about lady beetles a little bit separately, because they're not just coming for the honeydew. They're coming to give us some benefits. They're coming to actually eat the crepe myrtle bark scale. It's probably one of the most important natural enemies of crepe myrtle bark scale are these lady beetles. Now, with the lady beetles, we see multiple species. And not all of them, as you can actually see the one pictured here that has the dark color, are that characteristic orange, very typical ladybug look. The other thing I want to point out is that most of our lady beetle species have life stage that looks nothing like the beetle itself. In fact, on the right side, you can actually, if you look closely, you can see an adult lady beetle in there surrounded by a bunch of pupae. And that's what those teardrop shaped things are. That's the pupal stage. And so I understand that this is the pretty deep entomology, right? Asking you to identify life stages of lady beetles, but we've developed a resource, and I want to show this to you. We developed this resource to kind of help with this. So this is a postcard, and it's something that can kind of stick up in the visor of your truck. And we show the various life stages, like the males, females, and crawlers of crepe myrtle bark scale, kind of what an infestation looks like, their actual size in the smaller silhouettes. And we do the exact same thing for the natural enemies, these important lady beetles. We kind of give you what they would look like, as well as a silhouette of what their size would be. We can get these to you through the extension office, or even through, we can send some of these out to you if you want to use this resource. But we were able to get some funding to produce this to help with that ID part. What are they doing? Well, they're consuming a lot of crepe myrtle bark scale. And in fact, 70 to almost 90% over 10 days, if we flag a little branch and come back and look at them. So a lot of predation of bark scale is happening by these lady beetles, all the different species that are out there. We want to know if we could recruit them. It's a great idea, right? If we could ring a dinner bell, bring them to our tree and not have to spray it, that would be awesome. We've tried this with various scents. In the literature, the scientific literature, there are different smells that can attract lady beetles. We've tried it. It wasn't very successful for us. In fact, there are commercial lures that say they do that. We've evaluated them and have not had much success with them. What we did have success with is these yellow panels, or sort of putting some sort of yellow visual attractant to the tree. Now, I call them panels because we don't want to put a yellow sticky card out there, because the lady beetles will fly and get stuck to it. So these are non-sticky. They're made of material kind of like a political sign that's just painted sunshine yellow and out in the tree. And it does bring more lady beetles to these trees for predation. The main way, though, that most people are trying to get these populations under control are with chemical controls and insecticides. The majority of what's being used are systemic insecticides, the systemic neonicotinoids. Those are materials that we can apply to the soil. They'll be taken up by the vascular tissue of the tree and delivered to the scale insects as they feed. We also have some what are called IGRs, and those are insect growth regulators. Those materials can work fairly well, best targeted towards the crawlers though. And the systemic insecticides, as long as it's a feeding life stage, they can get a significant dose of that. But so we do have some different chemistries, some insect growth regulators, as well as some systemic products. Our two main systemic products that we have, dinoteffuron and imidacloprid, are the active ingredients, but you know those trade names: Xylam, Safari, Merit, Mallet, and a bunch of others. We do have some concerns about these. We know that there could be poisonings of some of our natural enemies, like those lady beetles, and we've seen this and observed it in the field. As you can see, the top left picture, those are larvae of mealybug destroyers that got poisoned after a treatment with mallet. And then we also have concerns whether or not these materials are moving into the flowers. Crepe myrtle flowers only have pollen; they're nectarless, but that could put those beneficial insects at risk. We did this experiment with a grad student, and we looked at the common timings, which are fall and spring, as I mentioned, based on the biology for crepe myrtle bark scale, with both imidacloprid and dinoteffuron applied to soil. And then we collected pollen off the trees the next year and assessed what was actually being moved into pollen. The bad news is, for both dinoteffuron and imidacloprid, they do move into pollen. We clearly have data that show that they will, at just label rates, move into pollen. And they will move into pollen at levels that could be harmful to bees. And that's what the LD50 information over on the side is. The good news, let's talk about something positive then. So we get the highest concentration if we do a fall application of imidacloprid. We get much lower residues in the pollen if we do a spring application. So coming into this time of year, if you want to use a systemic insecticide, you're going to put less of that material into the pollen if you're using something like imidacloprid. The opposite's actually true. You're still going to get a lot of translocation with dinoteffuron in spring applied. Now, you might say, well, wait a minute, that number in the fall looks really high. We think that's an anomaly. We had 6 trees we treated in this experiment, and only one of them showed up with a very, very high amount of dinoteffuron. So otherwise, all of the other 5 trees had nothing. It was non-detectable. So again, we think the way to go is, if you need to make a fall application using something like dinoteffuron, spring application, something like imidacloprid. And we can more effectively use these materials when we have to. We do have some alternatives. I've got a grad student and a number of them that have been looking at this product limonene, and you may know it as orange oil, Gooby Gone, has a number of different names. Smells great, by the way. But it has been used in the literature to control scale insects and mealybugs. So we put up this greenhouse test first, and it was like, does this actually work? So we were making, yes, weekly applications. I know how impractical that is, but I wanted to give it a fair shake. And really, that's what the label says. Go out and make weekly or biweekly applications of this material. It has very limited residue. So if we do that, we know that we can get really good control with something like limonene. As good control as you see with pyroproxyfen, that's an insect growth regulator. That's our distance or fulcrum material, or either one of our systemic materials. All right, the limit here, of course, is making a weekly application of this. But we still were able to prove that, limonene could be effective in this greenhouse study. Then my grad student, Jordan, wanted to take this to the field and look at it with landscape trees. And we're applying the limonene material again weekly, because we know at least that works, and compare it to some of these other things I've talked about before. And limonene applied weekly, and our panel treatment that I mentioned earlier, those yellow panels, actually work in a landscape level trial. This was basically a full year landscape trees around Auburn, where we were managing them with limonene or with panels and trying to keep the bark scale under control. And we did a fairly good job of this. If you look at the limonene line is the lowest one here. We did get a pretty good reduction with limonene. I really am amazed at what the panels did, which is the orange line. So just putting those yellow panels in the trees, bringing in natural enemies, a really good way to foster biological control. And then the standard insecticide is the blue line there. So you can kind of see that they're all intersecting, and we're really pleased with the outcome of this. We're repeating it again this year. And in fact, some neighborhoods are already adopting this, which is really fun to see people use your research. So what are our major takeaways? Well, crepe myrtle bark scale is major pest. We can use some tools like conservation biological control and limonene. I think we just need to use these wisely, and we could reduce our reliance on systemic insecticides. With that, I need to give a shout out to all the funding agencies that help support this research and my grad students. And then I can take any questions. I'll leave my contact information up there for the short term, and then I'll stop sharing and we can take any questions, do any Q&A that you have right now.
What is the life cycle and the time between instars when they're crawlers until they reach adults?
So it's all temperature driven, but the lab data that's been generated, we're talking about 2 months for them to basically after hatch to get to a point where they're adults. That doesn't mean that includes the extra time for the adults to then lay eggs, but that's just development time. So it could be basically 2 months or plus or minus depending on temperatures we have. But during the typical temperatures in the summer, that's what it would be.
Good deal. Thank you.
David, this is Harvey Cotton.
Hey, Harvey.
On your parts per billion, I'm just a dirt farmer, so my parts per million, it seemed like EPA said 25 parts per million was the lethal dosage. So to those, I was thinking that showed 2.5.
Let me go back to that. So I'm looking at the same thing you are. I don't memorize all of our data. So you're talking about these slides, right?
Well, I think so. I can't see them on my deal now.
Okay. All right. So you're talking about the residue slides though, right?
Residue slides. Yeah. What's our number that we're trying to get to that we won't cause harm to our pollinators?
So I think the contact and oral LD50s that I'm showing here, the days to reach that, this is based on what's called a hazard quotient based on honeybee. Right. It varies whether we're talking about honeybee, bumblebee, or even one of the other, native bees like the andrenid bees. But these days to reach contact LD50 being 2 days or 10 days or whatever it is, that's based on honeybee contact or oral exposure data. Yes. And I don't remember off the top of my head what the actual threshold was that is used to
in the early days, they kept saying it was 25 parts per million. And we were only finding in that early research 5 parts per million in our nectar and pollen. So we were feeling somewhat encouraged. Now we've had a lot more research going on since then. So
yes. Well, and so, the other thing that some of those early studies, they've got, depending on what they actually did, right? If they just fed it to them in a lab, it's one thing.
Sure.
Repeat exposure. What we're learning now is these honeybees in areas like Alabama, Texas, the South, where we overplant crepe myrtles, they are absolutely heavily foraging on these trees. The pollen rakes, and we have some work going on at our bee lab here. A lot of crepe myrtle pollen comes into the hives. And so now it's dosed over time, if you can think, right? If they were picking up just a little bit like they do in places like Brazil of crepe myrtle pollen, they probably wouldn't get the same dose as if with the heavy foraging we have that's going on here.
Gotcha. Okay.
So we're coming into this springtime of year, right? This is, let me go up to this. We're coming in, it's the end of March, April. If we come into springtime of year, if you were in October, we got to come up with something to do. You come across a treated tree. What are some of the options we talked about for finding a treated tree that's completely infested right now? What do you think you would do? because it's a very real. People are cutting down, cutting back their trees, and they happen to notice this little white stuff. They might call an arborist. They might call a landscape company, a contractor. They might call the co-op and say, "What am I going to do about this fungus?" A lot of times I call it a fungus on the tree, so you might hear that sometimes because it does look white and fuzzy. So they might think it's fungal. They think it's disease sometimes. But this is a very real scenario. What are some of the things you might do to react to a tree you find is infested right now?
Yellow cards and limonene.
Well, yeah, you could put out panels and you could treat with limonene, unless you're in a landscape where they have no tolerance for crepe myrtle damage, right? And then in that case, you might have to come in with one of these systemic insecticides, or perhaps something like fulcrum or distance to knock back those crawlers, or you're going to apply one of the systemics to the soil, just knowing that at label rate, you're going to have some sort of impact on bees. That's just collateral damage of that application. It's still legal, but it's still going to have that impact on bees.
Well, I guess I should clarify. I was thinking of it through the lens of the home gardeners, like we had discussed earlier, and trying to reach that audience. They're probably not going to have access to those systemics, or even if they do, we probably don't want them using them willy-nilly out there, right?
Yes. Well, it just depends on whether or not they're confident in calculating those rates. Those rates are either, written in terms of amount of shrub height or diameter of breast height. And I have trouble calculating, DBH sometimes. I can imagine just, my mom trying to calculate that to figure out how much of imidacloprid to put on a tree. That's not going to happen. So yes, the panels and limonene are much simpler interventions for the homeowner to do.
Okay, so could you review again for us? I know you said there was one chemical that was better in the fall and one better in the spring. Which were those? Yeah,
so they both work really well. I mean, we've put them out in our trials in spring and fall, and so the efficacy is fine. It's just if you're applying something like imidacloprid in the spring, we know we're going to get higher loads into the pollen. So.
In a commercial application, I mean, this was standard practice when I was in North Carolina, was to keep the blooms cut off to encourage vegetative growth. Would that be something that you would recommend to do if you were going to have to spray the imidacloprid or the other systemics during, in the nursery to keep it out of the pollen?
So that is a great thing if it's feasible, right? But when I look at some of the large crepe myrtles that are around, that's a challenge to do on some of the mature trees. I've even talked to our hort department about any sort of plant growth rate. I'm so used to talking about insect growth regulators, plant growth regulators. And apparently there's some research to use plant growth regulators to reduce flowering. So, there are other ways, I guess, to remove flowers. That's the best way. That would remove hazard altogether if there was no pollen for them to forage on. So,
hey, Dave,
yes.
There is a Florel or Ethifon is labeled to remove flowers. I don't know how much work's been done on crepe myrtles, but it has a label for knocking off flowers of certain species. Florel.
Okay. Yeah, and I have no idea how to use that dose wise. They'd have to talk to somebody who's smarter than me about that.
I smell a budding research project right here.
Something else that I think
absolutely right.
Could be interesting would be trialing azadirachtin as a contact insecticide that's got an anti-feeding component to it, so that we can stop that bug and see better efficacy in the landscape while not going to a systemic. Should be something worth trialing.
I've trialed it. We've had 3 years of data with that, and fortunately, it doesn't work unless there's some novel new formulation out there. We've looked at least 2 different formulations of azadirachtin, and neither one has given us the efficacy we've seen with something like limonene, because that is also a low residue material. That it doesn't have the residue that a systemic would. But yes, we just don't see the efficacy with azidiractin that we do with something like limonene.
Correct. As a standalone, I wouldn't think that you would.
Yeah. So the other things that are happening this time of year is pruning. We got a lot of pruning of crepe myrtle. I did have a grad student that looked at could they be successful if they were removed on a twig, and they can. We actually put them in a garbage bag and put them in a growth chamber and pulled them out a week later, and the scales seemed to be perfectly fine. Now they weren't sitting on the curb at 80-degree weather, but it's not 80 degrees right now either. So there is a really good chance that if those cuttings are laying at the curb, they could be a source of additional scales for the neighborhood, as that truck is sort of picking them up and flinging them around and driving them through the neighborhood. So something else to think about springtime of year, and what else is happening with infested crepe myrtles.
David, with that, do we think that much of this is spreading so rapidly because of prunings and mulching of prunings?
I don't have a good answer for that. I don't have a science-based answer for that. That has to be a component of it, simply because we prune so much off crepe myrtles every year. But I don't,
because the crop, I mean, I know there's a little bit of flight, but it's not like these things are flying like Japanese beetles.
No, and they don't even blow very far on the wind. So there's some work out of UT, and if you look at the distance they're blowing from an infested plant, and you put sticky cards at different distances, it's not very far. I mean, they could infest other trees in that landscape, but they're not blowing halfway across town.
I'm seeing in downtown Huntsville where a new crepe myrtle has not been planted in any of these landscapes, and they're eaten up with it.
So again, there could be movement of these on those cuttings, that are picked up and mulched. I just don't have I've not done the research to be able to say definitively that's what's going on, but it certainly is based on some of the other things we've looked at in terms of scale insects that are removed from living plants. Crepe myrtle bark scales removed from living plants.
Thank you.
Anybody else have any final questions? I don't see anything in the chat, and I don't see where I received any email questions in advance. All right, well, if no further questions, David, we greatly appreciate your time today, and we'll get this video posted on our website and up on our YouTube channel so other folks can go dig in and get that information.
Very good. Appreciate the opportunity to be here with y'all.
All right, thanks so much.
All right, bye.
Thank you.